All right, welcome to everyone coming in online and to everyone here in person. We'll wait just a couple minutes as people start coming in before we get started. All right, in the interest of everyone's time tonight, I think we'll go ahead and get started and get things rolling as the final people start trickling in online. Welcome again to our fourth session of Mini Med School this year. I am one of the co-leaders, Kevin Field, and we're really happy to have you all back online and in person. We have a really great talk lined up for you tonight. First, we will have one of the MD PhD students, Rebecca Rubinstein, to give the intro, and then followed up by Dr. Ransohoff after a short break. It is my pleasure to introduce Rebecca tonight. Rebecca is one of the fifth-year MD PhD students coming to us from Wellesley College, and she is right now doing her PhD in Sophia Becker-Dreck's lab in the School of Public Health, where Rebecca is studying breastfeeding and diarrheal diseases in a variety of countries. I think she is primarily based out of Nicaragua, but right now she is actually in the DR for another side project. So she will have a lot to talk to you about and is really a wealth of knowledge on this subject for anyone who wants to seek her out afterwards. Again, for people with any questions for the speakers, please put them in the Q&A box on Zoom. We will have people monitoring the chat and can read them out to the presenters as we're going. And for any technical support issues, please email us at our Gmail account, uncmedimedschool@gmail.com. And so without further ado, I'll let Rebecca go ahead and share her screen and we can get going.
All right, everyone, and I hope that we did test this, so give me one moment. Okay. Kevin, is everybody able to hear me from the speakers in the room so far?
Yep, I think we are good.
Okay, well, I will go ahead and get started. My talk is probably going to be less technical than Dr. Ransohoff's, but hopefully it'll give some helpful background info so that you could get the most out of his presentation. So thanks again, everybody, for coming to Mini Med School tonight for tonight's session on colorectal cancer. My name is Rebecca. Like Kevin said, I'm a fifth year in the UNC MD PhD program, currently in my third year of a PhD in epidemiology at the Gillings School of Global Public Health. Like Kevin said, my research is currently based in Nicaragua. I'm looking at genetically determined breast milk components and how they impact the risk of diarrheal infections and downstream effects of diarrhea in children. But I'm interested in GI health overall in both adults and kids. And so tonight is kind of a nice switch to thinking about a really prevalent issue in adult GI health. I just want to apologize that I can't be at Chapel Hill Public Library in person tonight. It looks like it's a good turnout though. I just want to give a brief overview of how a healthy colon works and what happens when we need to take it out before we learn about colon cancer screening and treatment from Dr. Ransohoff. You guys are really lucky to get to hear from Dr. Ransohoff today. I've heard him speak before about his career journey. He's had a fascinating and fruitful career studying colon cancer and developing tools to get better at catching it early. So you could not have a better authority on this subject. Let me see if I can advance to the next slide. Okay, awesome. We are good so far. So first, before we go into the colon, I just want to remind everyone that the colon is really the spotlight during the second to last phase in the journey that food takes from the mouth to when it leaves the body as stool through the anus. So prior to food reaching the colon, initially, as we ingest food, the mouth chews food and salivary enzymes start breaking apart starches. Then the pharynx helps swallow food to make sure that we don't choke on it, and the esophagus helps move food from the mouth into the stomach. Then the stomach muscle mixes and churns food, and highly acidic stomach juice with the enzyme pepsin starts to break down proteins. Then, with help from the liver, the pancreas, and the gallbladder, the small intestine absorbs most of the nutrients from food into the bloodstream and lymphatic system for us to use. And then finally, the food in mostly liquid form is ready to enter the large intestine, which is synonymous with colon. You can call the large intestine the colon or the large bowel as well. And here on out, the colon will really focus on water and ion absorption before the remaining food leaves the body as stool through the anus. And I want to go back. Hold on. Okay, awesome. So the colon is quite important for our health. It has three major classes of functions. First, it reabsorbs water and electrolytes from our stool for the rest of our body to use. This helps us efficiently use the water we consume and prevent dehydration, as we are constantly losing water through our skin and mucous membranes lining the openings of our body. So very important to get the most water we can out of our stool. The colon also forms, transports, and stores stool before it's ready to be pushed out, hopefully voluntarily at a convenient time for you when you're near a bathroom. In the image on the left, you can see that the colon is comprised of several mucosal layers and also two muscle layers that run perpendicularly to each other. One of them runs longitudinally and the other runs wide. And these together contract and help compact, move, and store feces through the colon. Lastly, another important function of the colon is that it houses the gut microbiome, which is one of the densest microbial ecosystems in the world. And it contains equal or more cells than all the cells of the human body. And we'll be going into each of these functions in more detail. So first, I was hoping that we could get this video to play. I thought it did a really good job of showing how, it looks like it is going. It doesn't have any audio to it, but it kind of shows us how the muscular layers, I did not want to go ahead, hold on. How the muscular layers work together to really push food through as stool. So you can see that the longitudinal and circular muscle layers, this is how that they're contracting together to move stool through the colon. And yes, you can see stool moving through right there. And as different parts are squeezing, others are relaxing. And those deep ridges are called haustra as well. And this all aids in making sure the stool can move efficiently through the GI tract. Somehow I find that video very soothing to just watch. It's almost like, I don't know, watching one of those soothing video channels on YouTube, even though we're talking about poop right now. Okay, so so the first function I wanted to go into in more detail is how the colon aids in water and electrolyte recovery. So as feces are being pushed through the colon also absorbs water and ions. Most people who treat and study the colon divide it up based on the direction that the colon is facing. So that's how this diagram is referring to different parts of the colon. Most ion absorption happens in the initial ascending part of the colon. Here, the body expends energy in the form of ATP to transport sodium away from its concentration gradient. And then chloride ions are also reabsorbed back into our body from the colon through this concentration gradient created by sodium transport. And then here, tight junctions or barriers that tightly connect epithelial cells in the colon to one another help prevent sodium and chloride from flowing back from our bodies back into the colonic lumen, or the hole that the tube of the colon. And because chloride and sodium are hydrophilic charged ions, they can also help draw water back from the colon into the epithelium as they are being reabsorbed. And I didn't know this before, but in doing research on the colon, I learned that we can actually absorb between 5 to 8 liters of fluid and electrolytes from the colon each day, which I think is kind of a lot. And then now, as the food is finished working its way through the ascending colon, it started as fluid form, but is now slightly less semi-fluid. And it's now ready to pass through the rest of the colon to become harder and more dehydrated stool. So now the stool, or the semi-fluid, is going to work through the transverse or horizontally pointing colon. It's pushed through by those circular and longitudinal muscles that we showed you in the previous video. And it's important to note that these muscles need to contract adequately. If they don't contract enough and the stool spends too much time working its way through the transverse colon, then then too much water can be reabsorbed, and this causes the feces to become too hard, which can cause us to experience constipation. So not good. And then once the stool has made it through, or the mush has made it through the transverse colon and over the splenic flexure, it's now ready to work its way through the descending colon. It's going to enter here in a semi-mushy state. And here water absorption continues until the mush becomes the consistency that we see in normal stool. The stool, very importantly, it has to be stored in the descending and sigmoid colon and rectum until we're ready to voluntarily release the stool from the rectum and anus. And if the motility here is too active or too excessive, we don't reabsorb enough water, and then we experience watery stool or diarrhea. So this is like physiologically why we might experience things like constipation or loose stool or diarrhea. It all depends on adequate muscle contraction and the stool spending just the right amount of time in each section of the colon. Okay, so the last function that I talked about before is the gut microbiome, which we have all heard about probably a lot in the past few years. So the gut microbiome consists of commensal bacteria, viruses, fungi, and protozoa, and I think helminths as well. The figures displayed here and most of what we know about the functions of the gut microbiome come from our studies of gut bacteria. They're the most understood and well-studied group in the gut microbiome. So bacteria have a smaller and less complicated genome overall and cellular structure than eukaryotes. Eukaryotes is a domain in biology that includes humans and all multicellular organisms. So because we're more complicated, I guess, than bacteria, this also means that bacteria can perform many more types of metabolic reactions to digest food than humans can. They can ferment carbohydrates, amino acids, mucus, and phytates that human cannot digest. They convert these into short chain and branched short chain fatty acids. And these metabolites and other metabolites produced by gut bacteria are very important to help regulating the host immune system and host metabolism. And they also help communicate with the nervous system through the gut-brain axis, among other functions. And I also just learned this recently. So this phytate metabolism that gut bacteria do, phytates are a form of phosphorus that are found in plants. They're not really toxic themselves, but if we have too many of them and they're not digested, they can prevent us from reabsorbing enough zinc and iron back into our bloodstream since they sequester zinc and iron. So it's very important for gut bacteria to break these phytates down. And then independently, the gut bacteria can also detoxify foreign substances and heavy metals in our body. And that's why they're considered to be part of something that we know as the exposome now, which is a way of conceptualizing the real, for example, in the case of like lead exposure, the actual amount of lead poisoning you're going to get from experiencing it in the environment. And then lastly, the gut microbiome helps us recycle and convert bile acids and amino acids. I just want to say that this is like probably a small sampling of everything that the gut microbiome does, and I'm sure that we're going to discover more functions of the gut microbiome in the next decades. So given all of those really important functions of the colon, you might be surprised to know that you can actually live without one given appropriate medical management. So some people need to have part or all of their colon removed. The most common reason is if somebody has colorectal cancer, which Dr. Ransohoff is going to go into detail. In some cases, it might be recommended for folks to have part or all of their colon removed prophylactically to prevent cancer, especially if they have high-risk genetic mutations for developing colorectal cancer. These genetic mutations include familial adenomatous polyposis, hereditary non-polyposis colorectal cancer, and Lynch syndrome. These conditions are all inherited in an autosomal dominant manner, which means that you only need to have one copy of the defective gene to have these conditions and to be at high risk of colorectal cancer. So this means that people with these inherited conditions will typically have one affected parent as well. And then sometimes, unfortunately, although I think this is happening less often now as our medical therapies get better, folks who have ulcerative colitis that is worsening or is unresponsive to medical therapy, you may also need to have your colon resected. And then another important cause of resection or an indication for colon resection is toxic megacolon. This is a condition in where the colon is inflamed and swells up, and this raises the risk of the colon bursting, which, as you can imagine, is life-threatening. So toxic megacolon is an emergency life-threatening condition. It can be caused by a number of things, including Crohn's disease or ulcerative colitis, ischemia or reduced blood flow to parts of the colon, cancer in rare occasions, and then also certain infections like Clostridium difficile and Chagas disease. So how do we actually remove parts of the colon? And I'm sure that Dr. Ransohoff is going to go into this in more detail, but I wanted to just give you a visual of how this might work. So it looks like there's several surgical options for removing parts or all of the colon. This first one that is on screen is total colon and rectal removal. And you can do this with a surgery called the J pouch. In this surgery, a surgeon will remove the colon and rectum, and they'll create a temporary opening or ostium at the ileum, which is the final part of the small intestine right before the beginning of the large intestine or colon. And this ostomy is there so that people can excrete waste safely while their body is healing from the surgery. And then once they're more healed, the previous ostomy, sorry, this keeps advancing. The previous ostomy is sealed off, and then the person is able to pass stool similarly to when they had the colon, thanks to the creation of a J pouch that the surgeon creates from small intestine. And at first, when a person is like adapting to having a J pouch, the stool can be very watery because this is the form that stool leaves the small intestine in. But over time, the J pouch actually like learns and adapts, and it learns to absorb more water, and the stool takes on a more normal consistency, which I thought was fascinating. I didn't know that small intestine had the capacity to do that. And then there's some other options. This one you guys might have heard of. It's called an ostomy bag or ostomy pouch. So sometimes if folks need to have part or all of their colon removed, they will have a permanent ostomy in which the surgeon creates a small opening or a stoma from their colon, which allows stool to leave the body in a pouch. And people with an ostomy pouch, it sounds really drastic and it is a major surgery, but they can still enjoy a high quality of life given that they drink enough fluids and follow appropriate dietary changes. Because people with an ostomy pouch don't have the benefit of a full colon to reabsorb water, they need to drink an adequate amount of fluids each day, and they need to compensate. And every time they ingest fluids that can cause us to excrete more water in urine and stool, such as alcohol and caffeine. Patients, there's a number of classes of food that can also cause patients to experience discomfort, gas, bloating, or changes to their stool consistency, They're quite numerous, these lists of foods, but for anybody that is interested, I really recommend that you check out these resources on the screen about ostomy dietary changes and just any kind of things you need to know as you're preparing for that surgery, or if you have a loved one or a friend that is going through it. You can find these links by searching for the University of Tennessee Medical Center colectomy site, the UCSF's ostomy education portal, and the Mayo Clinic ostomy link. And with that, I know that was pretty brief and surface level, but I'm happy to take any questions. I hope that this gives you a little bit of an overview to help us appreciate why the colon is so important, but how we might learn to adjust without one. And I hope you also enjoyed Dr. Ransohoff's presentation today. And I'm happy to take any questions. I might need to turf some of them to Dr. Ransohoff, depending on how technical they are. So thank you all.
All right. Thank you, Rebecca, for a really great warm-up talk. Are there any questions from people online? Of course, you can put that in the Q&A that Matthew and Nate are monitoring, and anyone in person can just raise their hand, like old school style.
Thank you so much for that talk. That was really great. I appreciate you going into such over the muscle layers of the colon. Now, this might be technical, but when it comes to the J pouch, is there like any voluntary control, or is that because you have different muscle layers?
That's an excellent question. And as I promised, I will probably have to turf that to Dr. Ransohoff. That's a really great question. Do you know, Dr. Ransohoff?
The muscle stuff happens above the J pouch and it's going to get the stool all the way down. When you get as low as the J pouch, the muscles are important. They're not the ones that are in the wall of the colon and contracting. But in the J pouch, you're down near the pelvis. There's lots of big muscles there that work when when we normally move our bowel. So the goal of the J pouch I'm not sure calling it voluntary would be best, but the goal of the J pouch is to have you be able to contain stool until you want to move it. And the mechanism is different than the muscles in the wall of the intestine. The goal of J pouch is to be a reservoir as opposed to having a bag that that just takes water and fluid, small intestine. Having said that, you should know that if you need an ostomy, there's football players and stuff who have ostomy. I mean, it's not as bad as it seems. One of the things that I want to talk about in a couple of minutes is needing an ostomy for colon cancer. It's maybe 5% of people, very small numbers. So you don't have to worry that if I have colon cancer, I'm going to have my colon taken out. That doesn't happen. And the reason that you need, and we'll look at this a little bit more, can you guys hear me in the metaverse or the whatever exosphere?
We can hear you.
All right. That colons have to be taken out is like Rebecca said, ulcerative colitis. You've got really bad inflammatory disease. That can affect the whole colon. You have to have the colon taken out. And the patients after that, the time you want to operate is when they say, oh my gosh, I feel so good. Now that I've had this terrible, I need all this medicine. Why didn't I do this sooner? Needing a total colectomy for colon cancer almost doesn't happen. What you might have to do is to take out the colon that's down by the anus and the rectum, because the colon, it's not lying free in the abdomen where you can, take out 6 or 12 inches and nobody cares. You barely notice it. If it's down where all the muscles are, you've got to dissect it out. And that makes it a big problem. So rectal cancer can require an ostomy or a J pouch, some kind of reconstruction. But most colon cancers can be taken out either endoscopically or by relatively easy surgery, often laparoscopic, and you don't know the difference when that happens. That's a long answer, but I really want to clarify. You should not think, oh my gosh, if I have colon cancer discovered, I'm going to need to have my colon taken out or have a bag. No. Am I clear?
All right, great. Any more questions from people in person or online? All right, everyone's saving them up for you. We will take about a 5-minute break while we change the room over for get Dr. Ransohoff's slides up. Thank you again, Rebecca. That was a really awesome intro talk, and we're really happy to have you here, even if from afar.
Thank you so much.
Am I okay?
Yes.
Testing, you can hear me?
And we can hear you on Zoom as well.
All right, everybody. Welcome back. And we are going to get right into Dr. Ransohoff's talk. And so just a brief introduction. Dr. Ransohoff was one of the first gastroenterologists also trained as a clinical epidemiologist. He has a longstanding academic interest in improving methods to evaluate screening tests for cancer. He graduated from Case Western Reserve University School of Medicine and completed his internal medicine residency at Dartmouth College. He did his clinical epidemiology fellowship at Yale University and his adult GI fellowship at the University of Chicago. As far as colon cancer screening goes, he has evaluated colonoscopy, sigmoidoscopy, stool blood tests, and more recently, evaluated molecular markers used in colorectal cancer screening. He has served as an advisor to the National Cancer Institute, the FDA, and the U.S. Preventative Services Task Force, and has offered and to other organizations and companies as well. He has also served in editorial positions with numerous journals. At UNC, he has led faculty development programs for junior faculty and fellows as director of the Robert Wood Johnson Clinical Scholars Program and of the NIH-funded training program. Clinically, he sees patients with general GI problems, and we will hand it off to you.
Good, thank you. Can you guys hear me? You can hear me over there? And Kevin, remind us about the rules for people in the room. If you want to ask questions, can you raise your hand or something? Is that what we want to try?
Yep, so for people in the room, you can just go ahead and raise your hands with questions. And for the people online, if you put your questions in the Q&A, one of us here will raise our hands for you so that your question can be read out.
I'm happy to try to answer questions as we go along. This is actually the first time that I've ever given this talk for a sort of lay audience and an overview and so forth. So this talk may have more in it than I can cover. I will do my best, but I'd like to try to answer questions along the way. If it gets out of hand, we'll change the rules. Okay, so we're going to talk about colon cancer. There we go. Disclosures. And so the goal tonight is we're going to do a colon cancer overview, colorectal cancer, that's CRC, for a non-medical audience. And this is sort of a big area. And it was fascinating for me to think about all the things that I could talk about that are really neat that we don't have time to go into detail about. But I've selected some topics in depth, like what's actionable? Does diet make a difference? Is screening important? What do people want to know about? What about the microbiome? And then there's a couple of illustrations where I want to go into some depth about how science works and how we've generated knowledge, some about screening, some about molecular markers like the Cologuard test, that sort of thing. There are really some fascinating adventures. Why has colonoscopy become so popular? There's some really neat stories here that in medical school or, as normal human beings, people may find interesting. So we're going to talk about what is colon cancer, what are the risk factors, and is it preventable? What about screening? That's going to be really important because the main message here is screening is a really big deal. If there's one take-home message, we can talk about physiology and diet and all this other stuff. Screening is important. We'll talk a little bit about blood tests, surveillance after polypectomy, and we could talk about all these with some fun in depth. But first, Rebecca mentioned my career, and I just wanted to tell you a tiny bit about it because it's interesting, at least to me. My interest is in clinical decision-making and evidence generation. This is the field of clinical epidemiology. It's where you apply the tools of epidemiology to clinical decisions like diagnosis, what makes for a good research study to understand whether a diagnostic test works, how do we understand prognosis. These are fields different than normal epidemiology. We call it big E epidemiology, like in the School of Public Health, where they're mainly interested in etiology, but there's a science of clinical decision-making. And where'd this come from? As a child, my father and grandfather were general internists in Cincinnati. I was conceived, born, and raised to practice with them. but I didn't. And my dad used to show me difficult decision-making issues. He'd say, well, I saw a patient today, they got a sore throat, they want penicillin. And Dad said, but most sore throats aren't caused by bacteria. Penicillin doesn't do any good, and penicillin can hurt you. There's 200 penicillin anaphylactic deaths in the United States every year. And so treating a sore throat with what the patient wants may hurt them without benefit. If it's viral, you have an anaphylactic reaction and die, you got no benefit and you died. And Dad was thinking about, you got a patient in front of you, what's the benefit? What's the harm? Where do we get the evidence for this? How do you make a good clinical decision? And then how do you explain it to the patient? This kind of problem applies to so many things that we're faced with now. It's a whole big topic of its own. But what my dad was telling me about was clinical epidemiology. It didn't have a name back then, but that was really interesting to me. So I trained in med school, and I asked could clinical judgment be taught to non-medical, to non-MDs, because when I was a kid, there was a doctor shortage. Can you believe it? And after medical school and residency, I found that there'd been a book written called Clinical Judgment by Alvin Feinstein, director of Yale's Clinical Scholars Program and a godfather of the field of clinical epi. And let me get rid of this. You got to see the top. Anyway, this is the Clinical Scholars Program. Here's Alvin, the director. Want to guess which one is me? That's me. And my hair looks a little long, but boy, it was really long before I got to Yale, because I'd been in New Hampshire at Dartmouth for internship and residency. And every 3 months, I lived on a farm renting a room, and in West Leb, New Hampshire. And every 3 months, I'd go to the barn, set up a mirror, and cut my hair myself. But I mean, my hair was all over the place. So, this was a change. Ralph Horowitz became head of medicine at Yale and then a dean. And back then we all had long hair, and we were all different drummers, and we were all worried we'd never get jobs because clinical epi had no home. This was different drummers. Every day at lunch, we worried about having jobs. Now it's become unexpectedly mainstream, and I did a GI fellowship at the University of Chicago, which was one of the major medical GI centers in the United States at the time, and have applied clinical epi since then. More to say about lessons from this, but this was an adventure, and Alvin was great. He was a genius. He was a little hard to work with, but he was a genius. So we're going to talk tonight about what is colon cancer, and that's where we're going next. Rebecca's told us about anatomy. This is the colon and rectum, and we've talked about the different parts here and stuff like that. And by the way, the blue slides, this is the American College of Gastroenterology. I took a lot of their slides and included them, and I'm going to make comments on them. So thanks to that for them for this slide and some others. This is the colon lies in the abdominal cavity, and it's relatively easy to get to if you've got surgery. And if you've got a cancer in the sigmoid colon, you can go in and even laparoscopically and take out the lesion. Sometimes it can be done endoscopically, but you'll not notice much of a difference. You can live without half your colon. It absorbs water. We've got lots of people walking around that have half their colons because of the blood supply. Sometimes it's easier to take out half the colon, but that's not a colectomy. You don't have a bag, and you can have pretty normal bowel movements. And if you take out less of the colon, it's even less of a problem. So please don't be worried about ostomies and colectomies. They happen, but they're rare. If you look down here in the rectum, what's happening is you've got all the muscles that when you sit on the toilet and you squeeze and things like that, these are the skeletal muscles, the striated muscles that contract and so forth that you control. And the colon is sort of directly attached to that. And if there's cancer, you're messing around with these other muscles and so forth. The surgeon is, and that's why you may have to have a J pouch or something like that. But otherwise, you don't. Everything stays intact, and you just take out part of the colon. So don't say, oh my gosh, if I get screened and I have colon cancer, I've got a colectomy or something, or an ostomy. You don't. That's really important to understand. So the main function is to absorb water. We don't need all of it Digestion and absorption happen, as Rebecca said, higher up, mainly in the small bowel. The bacterial cells or the microbiome in the colon outnumber the cells in the body. Did you know that? And the microbiome likely does critical things that we do not understand. Stay tuned. I think this is going to be a really big deal. And so as long as we're talking about the microbiome, let's just say a little bit more. What is it? It's bugs in some place. Microbiome in the colon. I've got a friend who studies the microbiome in soil. You pick up a teaspoon of soil, there's zillions of bugs, and they've all got relationships with each other, and they help each other and hurt each other. And I mean, it's just, you know, it's everywhere. We're just talking here about the microbiome in the stool. And the stool microbiome likely has important effects. Like in weight, if you take the microbiome of a mouse, pardon me, if you take a mouse and change its microbiome by implanting bugs and stuff like that, and give it the same diet, it can gain weight or lose weight depending on what you give it. This is mind-blowing experimental stuff. There's experimental evidence that diseases like diabetes may be related to what's going on in her gut. Immunological problems, rheumatoid arthritis. I'm not saying they do. This is the kind of thing that's being studied. Cancer may well be affected by the microbiome, cause of. But the caution is, or you know, the situation is, the importance of the microbiome may be huge, but we don't understand it enough to do any intervention. So if you start seeing ads about probiotics and microbiome and stuff like that, this is all unproven stuff. I think if we could come back in 50 years and see what was going on, we are going to know some incredible stuff about the microbiome, and it may be really important, but it's so hard to study. And one of the reasons why there's no structure to it. More cells than in the body. Well, how do we study the body? Well, we got organs. We've got eyes, we've got digestive system, we got a brain, we've got muscles. You know, we can study tissues and organs. In the microbiome, it's all just one big gamish. And there's lots of different kinds of bugs there. And we just don't understand. It's just really, really hard to study, but it's going to be important. And for those of you who are sort of philosophical, it may turn out that we were put here on Earth to carry around the microbiome so it could do stuff. And I'm not totally kidding about this. You know, when you think of birds and carrying seeds and other stuff, maybe our job is just to carry the microbiome around the world, or who knows what. I'm sort of teasing a little bit, but not completely. This is going to be a really big deal, but not in some of our lifetimes. All right. So let's get back to colon cancer. It's a big problem, about 130,000 cases a year, 50,000 deaths. It's the #2 cancer killer in this country. Lungs #1. Breast is important in women, but you know, colon affects men and women. It's really important. Adenomatous colon polyps, which we'll talk about in a minute, are the precursors to colon cancer, and they're very common. 30 to 50% of people over age 50 have adenomas. We'll talk about that in a minute. And because we've now got high definition scopes, and because doctors get rewarded for finding adenomas, do biopsies, make a diagnosis, that gets a medical bill, you get judged on how many polyps you find. I'm sort of exaggerating a little bit, but not completely. We find these tiny things that we call ditzels. They're about the size of a grain of rice. Most are tiny. To remove them all is a big job. Which adenomas will become colon cancer? That's what we want to know, and should be targets and be removed. It's going to be mainly big ones that have certain kinds of characteristics on biopsy. And then this is really important. Which adenomas after removal? The adenoma is gone, but which ones indicate you got a high future risk of having more adenomas and cancer, because that says, do you need colonoscopic surveillance? Most of the people in this room are too young to have even considered that. Some people who've had colonoscopies may know, or in the audience, you know, online, if you've had polyps found, you may get recommended to come back. Which adenomas after removal indicate high risk in the future? That's a big deal. Most polyps are ditzels of low importance. Oh, I thought you guys were scratching your head. Are there questions over there? No, this is fine. We're at the bottom of the slide. Yes, sir.
About 130,000 cases of bullets with cancer each year. Is that in the US or?
Yes, I'm sorry, it's in the US. Yeah. Anything else? Nope, okay. Any other questions here? Comments? Nope, okay, all right. Okay, so let's look at this adenoma stuff. And what we're looking at here is as you go from normal tissue to adenoma to colon cancer, phenotype, that's the anatomy, what it looks like, follows genotype, what's the gene? So here we're looking at, this looks like a little piece of chocolate to me. My wife is a professional chef, so, you know, I think along those lines. This, or, you know, it could be a jelly thing with a couple of layers. The purple stuff on the top, this is the inside of the colon. And this is the part that absorbs water. It also secretes mucus and stuff like that. And then underneath this yellow thing is the submucosa. There's a tiny muscular layer here called the muscularis mucosa. And then there's a bigger muscular layer. This is the one that contracts and stuff on the outside. So the top layer is where the action happens and where adenomas happen. Adeno means gland, oma means growth of a gland. So here's normal tissue. And then you go to this next step. This is a little polyp. Polyp is Greek for bump or something like that. And you can see here, you can see here a bump. And as you look down here, here's the chromosome changes, the genetic changes that may drive what is going on. And then as things progress, this is a bigger polyp and it's on a stalk. So he's going to be easy to take out. And what do you see here? The purple stuff, this is glands. And this is sort of, you know, this is relatively normal glands, but as the polyp gets bigger, you see this dark thing here? This is cancer. It's cancer histologically, meaning in the tissue when you look at it. It hasn't invaded yet, but it looks like cancer. We could call it carcinoma in situ. If it invades, that's a bad thing. And what's happened, but you know, if you do a polypectomy here, you've cured it. And even if this thing invades into the stalk here, if you take it out, it's likely cured. Colon cancer doesn't spread easily. That's one of the nice things about it. But when you get down here, and you know, these other chromosome changes have happened, P53, let's see, APC is going to be up here and so forth, adenomatous polyposis coli, that gene. Here, this is normal adenoma, but you see all this blue stuff, and the blue stuff is, that's what it's looked like under the microscope. The nuclei is colored blue, the nuclei get large, the cells lose their sort of structure like, like this tissue, and you can see how this is invaded. This is invaded through the submucosa and actually through the muscle wall. This is a bad thing. And what this orange thing is, this is the liver. So, you know, after it gets out and it goes to the lymph nodes, it can travel to the liver and so forth. But the implications here, early discovery can cure. This is possibly totally curable. This definitely is very curable. And this is what it really looks like under the microscope. So the colon lumen, the tube of it is at the top. On the outside, you can't see it real well, but this is the serosa, this is the outside, and this red stuff is mucosa here. And if you look over here on the left, these things are glands, and they look a little bit like test tubes. You can see that. And then this is the muscularis mucosa right underneath, which, you know, when that gets invaded, that's a kind of cancer. But as you move over here, what you see is the test tube-like features are gone, they're leaving, and there's a lot of blue here. These are a lot of nuclei, and the cells are doing things we don't like. They're invading. And you see here how this is sort of a muscle layer. This is the muscularis mucosa. It's gone here. And we've got these blue cells that have invaded, and this is bad. This is an invasive cancer. Now, this guy's, you know, likely totally curable because it hasn't gotten to this muscle layer where it's more likely to spread. But this is what cancer looks like. Here's more cancer on the surface. This is, you know, this is likely cancer here. You get back over here, we're back to normal cells again. Normal adenomatous glands. So that's what it looks like. Polyps come in different shapes. This is a polypoid thing. Pardon me, this is a polyp on a stalk. This is very easy to take out completely. This guy here is what we would call a ditzel. This is a relatively big ditzel. It's very, very small. The vast majority of polyps that you have when you have a colonoscopy are very small. Small means under 0.5 millimeter, or in England, small means under 1 centimeter. That's 10 millimeters. We get scared about that in the United States. In England, they don't. And they've got some evidence that even people with 1 centimeter polyps, if you take them out, they don't have a higher risk of cancer in the future. In the United States, and this is a whole interesting another story, we are sort of terrified of even ditzels. And we've trained ourselves and the public to think that any adenoma, any polyp means don't buy any long playing records, no magazine subscriptions. It's gonna, you're in big trouble, and it doesn't work that way. This is, this is another polyp. These are two. These might have cancer in them. They probably don't. This is, this is the normal lumen, the normal tube of the colon. But this is a polyp. And let's see, this is the normal lumen. We're probably looking at the ascending colon right here, looking up. And some polyps are hard to see. These actually aren't that hard to see. This is a polyp. And what's going to be hard is how far does he spread to either side? Some polyps are really, really flat. We learned this from the Japanese about 15 years ago. And polyps on the right side of the colon, the ascending colon, tend to be flatter and harder to find, and that presents interesting problems in screening. This says some polyps are hard to see. This isn't hard to see at all. Look at this. See this black thing? That's the scope. It's turned back on itself. It's in a U. It's looking back, and that's how they discovered this guy. And then, here's things that I'm not going to go through, but there's a lot of bad actors on here. I do want to show you this. You see this one cell here? You see all these sort of polypoid things, the entire wall of the colon. This is probably something, this wasn't labeled, but this is probably somebody with adenomatous polyposis coli that Rebecca mentioned. It's a syndrome where you have a mutation in your germline, and you will get cancer by the time you are age 30 and have a colectomy for that. It's very rare. And we can't treat it medically. And basically the whole colon is like a big polyp, and the colon's 5 feet long. And, this is a lot of places that can develop cancer. This is very, very uncommon, but it's just showing you, what things can happen. Most of these other people are, these are cancers here. This is an ordinary dull polyp on a stalk, and you can see the wire here. This is going to go over the polyp onto the stalk, and it's going to be easy to take off and get cured. This is not likely at all to have cancer in it. If it does, it's going to be cured because it likely hasn't invaded at all. Yeah.
Generally, how long after
A few days. So getting back to big picture, risk factors doubles with each decade. Family history, the syndromes are important, like Rebecca mentioned, familial adenomatous polyposis or adenomatous polyposis of the colon, APC. The mutations happen, and this is really bad. Hereditary, and you have to take the colon out for that. Don't worry about it. It's really, really rare, and the doctor's not gonna. It's very unlikely that somebody, would just discover this at a screening exam. And actually, most people present with problems by the time they're 30, or they have a family risk and get worked up. But this is not going to get discovered at age 50 when you go in for your colonoscopy, or 45. Hereditary non-polyposis colon cancer. This is something that happens on the right side of the colon, and it's associated with other cancers. Rebecca mentioned colectomy for this. Many people don't need a colectomy. You do need a colectomy for FAP, but that's really rare. HNPCC, there's fewer polyps, but they can be bad, and they can be more common, but it's not like the whole colon is an adenoma like an APC. But these are syndromes that are uncommon. Sporadic cancer, which is most common for all of us, has no strong genetic markers. And so we look at phenotype, first degree relative. If your relative has colon cancer, risk increases but it depends on the age of the relative. So when relatives have colon cancer at age 60 or 50 or 40, then we start getting worried. If your grandmother had colon cancer at age 76, that really doesn't do much for your risk. Pardon me, if your mother did. We don't know about grandparents. For breast cancer, we understand, aunts and uncles and grandparents and what that all contributes to your risk. For colon cancer, we don't have that fine detail. It's going to be first degree relative, mother, father, kid, sibling. Younger is important. Question?
Yeah, so, question from the chat. This, I think, refers more towards the previous slide. But as, before to know if a person has a polyp removed during colonoscopy, they can go home and eat.
Oh, yeah.
Why is that? Does the removal cauterize that?
Well, yeah, for big polyps, we use cautery. For small polyps, we just scrape them off with a cold wire now. And the reality is that most people just don't have problems. If you're going to have a problem, if there's a big stalk, for example, and it's got a blood vessel in it, the doc will do cautery, and they may warn you, we don't really know what prevents, but don't do a lot of heavy lifting or just be on the lookout. But you don't go home with instructions not to eat. You may just get instructions to sort of watch and don't do real vigorous exercise for a little while. But the vast majority of polyps, just, there's nothing that you got to do. You can go home and just resume normal life. Yeah.
So the cancer is not invading.
Yeah.
It's seen when we get
I'm not sure exactly what you mean. If a cancer, yeah. Oh, yeah. Oh, yeah. If it's a small polyp, the chance that it's got cancer in it is just about zero. If it is a large flat polyp, that can require more maneuvering to get to it. You inject some fluid underneath, you try to lift it up, and then you scrape it off and stuff. And then that gets taken to look at under the microscope.
Is there the risk of issue bleeding out?
It's uncommon, but it can happen. Usually it's not bleeding out, but some people do, and it happens, as you get old. I mean, having bleed where you need a hospitalization or a transfusion. It's a very low percent. It's worse as you get older, but we do zillions of these, and most people do fine. But there are rare instances. If you're on blood thinners, that's a big deal. Most of the time, bleeding is not a problem. It can happen. It's more with bigger polyps. I don't know if that answers all your questions, but pardon? Okay, all right. We can talk later too. Okay, so let's look at factors that further increase risk. Family history, we've talked about. Family history of adenomas, if we just took all adenomas and anybody with a family history, everybody in the world, this is like the ad that says, do you know, was your relative, and were they in World War II? Do they have a friend who was in World War II? Do you know anybody who knew about World War? It's sort of, everybody in the world qualifies, and we don't act like adenomas are a risk factor. If somebody's had large adenomas, that might be a risk factor, but as docs, we can't use this because most people, when you ask them, they don't know about their own history of adenomas, much less a relative, the important information. Size, number, histology under the microscope. They just say they had polyps. So we don't really use this. Cigarette smoking increases risk, about 15 or 20%. Obesity increases the risk of lots of cancers, and as well as heart disease and diabetes. There's a whole lot of reasons for worrying about obesity. Gallbladder removal is not very important. Physical inactivity, a little bit of importance. Abdominal radiation, yep. Cancer of the uterus and ovaries before 50, this is Lynch syndrome. And Lynch syndrome is associated with cancers in multiple organs, because the biological problem is not something in the colon cancer pathway. It's a problem in a DNA repair pathway. And when DNA doesn't get repaired, then cancers can arise and can happen in multiple cancers. There's a great story about how that got discovered. Ulcerative colitis, yep, big deal. African-American race. Which factors are actionable? And the answer is some, smoking, obesity, physical inactivity. Some of these things are actionable, but it can be really difficult. And by the way, for obesity, your motivation shouldn't be to avoid colon cancer. We have a terrible obesity epidemic in this country. The obesity causes lots of other diseases. It's a gigantic problem. Fatty liver, cirrhosis, diabetes, heart disease, cancers of multiple types. But, treat obesity for other reasons, and it can be hard to do. This is the most important lesson for the whole night. The most important risk factor is not getting screened. All this other stuff is too hard to do for a lifetime to change my diet. You might want to do it, and for a variety of reasons, but the neat thing about colon cancer is when I started my career, we didn't know that screening worked, and I thought this would never be worked out in my lifetime. We know that screening works. We'll talk more about it in a couple of minutes. Not getting screened is the biggest risk factor. Should I have people repeat that after me? This is really important. Yeah.
Is there a simple time when passage or follow-ups develop? Like, is it possible that one colonoscopy didn't come to and then another colonoscopy a few years later, and then you try to see this?
Sure. Well, you can see little things, when you've had a colonoscopy and it's clean, there's two things you got to consider. Did I have a really good prep? If you ever have a colonoscopy, you have to get a good prep. It makes the exam easier. And then we know if there's nothing there that you've got a clean colon. If you don't have a clean prep, we can't tell, and stuff can hide. So a polyp appearing later, or even a cancer, could have been something that got missed before. But in general, the growth of adenomas is slow, and we're talking about many years. But yes, they can occur. They do occur with older age, and that's why we do screening over time. Yes.
Why or how is gallbladder contribute to this?
I don't know. And gallbladder has actually been one of my hobbies. It could be, it'd be a change in bile acids, because some kinds of bile acids, lithocolic acid, can be carcinogenic. But this is not, we don't make screening decisions different on people on the basis of gallbladder removal. I think if there's an association, I do not think it is very strong. If somebody wants to correct me and look it up, let me know. But this is not, the task force and the American Cancer Society don't say, like, for family history, you got a family history, we're going to change the recommendations. Nobody does this for gallbladder removal. Anything else?
I
Okay.
did have one question, Dr. Ransohoff. Can you
All
hear me
right.
over Zoom?
So can you prevent this
Oh.
by diet or by aspirin and other non-steroidals? Yeah.
That could have a question. It looks like it's not able to
Unmute properly. There's something up. I guess I'm not plugged into the speakers now. Is it known if the association with African American race, is there a genetic component to that, or is this like through like systemic racism or some other factor?
It's complicated, and I'm actually not sure whether that refers to something biological about cancer is more common or harder to treat, or it has to do with issues of access to care and other things, which could be related to racism and so forth, and access. And so I don't know the detailed answer to. I don't really know exactly what that statement was referring to. Anything else? Okay. So can you prevent cancer, colon cancer by diet? We haven't talked about aspirin. Aspirin, you remember, a few years ago, people were saying, take aspirin, baby aspirin, we can prevent heart disease, we can prevent colon cancer and stuff like that. There's some neat mechanisms how medicines like aspirin, they're non-steroid anti-inflammatory drugs. Aspirin is non-steroidal. Other non-steroidals are things like Advil and other kinds of medicines like that, Aleve. And they can prevent, they can interfere in the pathway that leads to colon cancer, and they've both been studied. Diet can contribute, like red meat and fat, but the association isn't strong. It's very hard to study. And any intervention is very difficult and long-term if you want to intervene with diet. I'm just making the case that, you know, this is sort of interesting biology, but it's not very actionable. This is a different issue. Aspirin and non-steroidals may prevent adenomas, and the MCI got really interested in this. Boy, we've got a mechanism, we got a pathway, we can prevent adenomas, maybe we can prevent colon cancer, but it's really hard to study, and this is a big deal. Side effects, even if they're uncommon, can outweigh the benefit when prescribed to healthy people. For example, aspirin can cause serious bleeding and a little bit of GI bleeding. You know, we don't worry about that much. Most people don't have problems with it. You bleed into your brain, and that can be a big problem. And it's one of the ways you can have a stroke. And here's two articles that you can't see totally about why. I don't know. I don't think we can get rid of the stuff at the top. These are articles that came in part from UNC. Dietary calcium has limited benefit to prevent adenomas. Calcium supplements were studied in a clinical trial, New England Journal. John Barron is here. He was at UNC. He is at UNC. And so the idea was calcium, we think, can prevent. Let's study it. Huge study. Had a relatively negative result, meaning there was a tiny benefit, a small benefit on adenomas, and we didn't even know if that's going to affect colon cancer. And big study, expensive, interesting question, important. It's in the New England Journal with a negative result because it was an important question, but it's frustrating. Here's another study from UNC with Bob Sandler, who is head of GI, John Barron again, non-steroidals, rofecoxib here, has side effects of cardiovascular things. I think it was heart attacks here. The point is, if you've got a drug, it's got a small rate of a bad side effect, but if we're going to give this to millions of people, we got to be sure that it's really, really safe. It's a high bar. The point is that diet and non-steroidals as prevention, it's a difficult climb. So I wish we could get rid of this stuff on top. Go away. This is, oh, you think?
That next image, we're up there on the top right, I think you should go to the floating media controls. And then you can click and drag your talking panel somewhere.
This is why we have younger people here. I should have known that, but thank you. See, I've learned something tonight. All right. Can symptoms identify colon cancer? The answer is no. Should I wait until symptoms? And remember, this is the ACG. Don't hesitate to talk to your doctor, blah, blah, blah. Blood in the stool. Most of the time, that's not colon cancer. The vast majority. There's things like hemorrhoids. There's things like AV malformations. Hard stool can cause tears in the skin and stuff like that. We see so many people in clinic who are referred by their docs and they've got some bleeding and they're terrified they've got cancer. And, you know, for most people who come in with, you know, just intermittent bleeding, that, you know, the chance that you've got cancer depends a lot on your age and your family history and stuff, but it's, you know, it's a tiny percent. Most of the time it's benign. Don't be afraid to talk with your doc about bleeding if you have it, and don't get overly worried about colon cancer. Change in bowel habits. I thought this was neat. This is the ACG. They don't know how to spell. Change in bowel habits, that's pretty non-specific. Shape and size of the stool, ditto. Rectal pain, you know, if rectal pain is caused by colon cancer, you got a big cancer down there. Most of the time cancers just sit there and they bleed, but they don't cause pain. Abdominal pain, much too vague. We see zillions of people with abdominal pain. The way you get pain with colon cancer is like if you have an obstruction, and that's a late stage thing. Abdominal pain is not useful for it. You know, it's not indicating colon cancer. Unexpected weight loss it's got to be worked up and by a general internist. Colon cancer is in the differential. Depression is, thyroid disease, lots of other cancer. It's a really broad differential, and usually you can figure that out by history. Unexplained or new anemia, I put an arrow here. This is a big deal. Anemia in an adult man or a non-menstruating woman is colon cancer until proven otherwise, if it's iron deficiency anemia. You really have to worry about it. Colon cancer usually doesn't bleed in a way that you can see it. It oozes. It doesn't bleed red blood. It can, but not often. usually you present with an anemia where you say, I had no idea that I was passing any blood at all. So an anemia has to be thought about carefully. Some symptoms are important, others are neither sensitive nor specific, but don't wait for symptoms to get screened. That's sort of the bottom line. So let's look at how good is screening. It's more effective than for other cancers. Did you know that? Here's the cancer. Colon reduces mortality by 30 to 60%. Is there evidence from a clinical trial, which we like? Yes, and it was positive. Mortality reduction. Why? Because it grows slow and it metastasizes late. Breast cancer, we do mammography, but the mortality reduction is only 15 to 20%. We do it, but numbers wise, it's less effective. Yes, we got clinical trials, they had positive results. Screening's not great. We do it. I mean, it does work, and that's important. Treatment has been better for advanced disease than in the past. That's one of the reasons people survive more with breast cancer now. Lung cancer, yep, mortality reduction from lung cancer, 20% reduced by the low-dose CT exam. Yep, clinical trial, really big deal. Cervix, real high mortality reduction, and there's never been a clinical trial. This is a one-off where screening got adopted without a clinical trial because Papa-Nicolau invented the pap smear that got adopted in the 30s or whenever it was, and the temporal trend was so dramatic over time that we didn't need a clinical trial. This is never going to happen again, but it did for cervical. And the cervical cancer is nice because the organ is small, you can see it, on exam, you can stain it, and you can sample it to see is there dysplasia or early cancer there. And now we've got HPV tests and stuff, and it spreads very late. So this is a one-off. Ovary, there's no mortality reduction. And we've had two great clinical trials, one from the NCI, one in England, and the trials have been negative. And the reason is because ovarian cancer is really sneaky. It metastases because of where the ovary lives in the abdominal cavity and surrounded by the fallopian tube. The cancer begins in the fallopian tubes, not in the ovary. And the fallopian tube is connected. Cells can float to the rest of the abdomen. So by the time cancer is discovered, it's often metastatic to the abdominal wall, to the wall of the abdomen. And it's a terrible disease. It's a huge problem, but there's no evidence of mortality reduction. It's been really so frustrating, a problem. Prostate, low mortality reduction. The task force gives it a grade C, which says, we're not recommending it. You can talk with individual patients. The problem is many cancers don't do harm. They got a label of cancer, but they don't progress and kill you. And the side effects are important. Double-digit rates of impotence and incontinence in the past, especially for prostatectomy. And one of the paradoxes is this is sort of like my dad with penicillin and sore throat. You operate on people, they're impotent and incontinent afterwards. You call them on the phone. Some people at Dartmouth did this and published it in JAMA. Call them on the phone, they're survivors, and you say, would you do it again? And they would all do it again because they all think they're survivors. The vast majority may not have been helped because many cancers don't go on to kill people. It's a complicated thing. It's a really interesting phenomenon. Okay, so screening recommendations, who and what age to start and stop. This is from the task force. Look at the middle here. Pardon me, look at the top. Adults age 50 to 75 years, it gets an A grade. Everybody ought to get screened, period, the end. This is a big deal. We lower the age to 40 to 45 to 49, it gets a B. When you get older, age 76 to 85, the task force says, nope, don't do it on everybody. Figure out, do they have a life expectancy that's long enough, like maybe 10 years, that it's worth doing. So this is about starting and stopping. Yes.
I guess you're saying.
You mean like in an older person?
Yeah.
If, that when you discover cancer, it may be years before it would go on to kill you. If somebody dies with colon cancer and they've never been bothered by it, who cares? That's the idea behind it. Now, if you're 75 and you're planning to live to 110, then, then get screened. But if you're 75 and you've got bad heart disease and bad diabetes and so on, make that decision. And as you get older, the risks of colonoscopy, they're not very high, but they're there. That's something you've got to think about. And it's, the task force is just saying, think about it. They're not saying don't do it. They're saying don't do it automatically. If you're 50 to 75, they're saying do it automatically. So what tests are there to do? There's non-invasive tests that are triage. And if they're positive, you do a colonoscopy. If they're negative, you don't do a colonoscopy. Stool, fecal, immunochemical testing. It's looking for blood. Fecal, immunochemical test for blood. Stool, multi-target DNA test. This is Cologuard. It's multi-target DNA with the fecal immunochemical test. These are the non-invasive tests that people tend to do. Blood tests may be in the future. Colonoscopy finds and treats, and it's done to work up a triage test if it's positive. That's the main use of it. That's one reason you know colonoscopy works. But it can also be a primary screening test that's common, like in the USA, it's very common, but it's not preferred, according to the task force, in spite of promotion by gastroenterologists, because programs of triage testing over time are equally good. We'll talk about that in a second. If you get a colonoscopy, do a good prep and a procedure by an experienced person. Again, have I made that message clear? I want you to repeat after me. You're going to be quizzed when you leave the room. You have to do the prep. Don't say, ick, I'm not going to do it all. You want to be whistle clean. Okay. Screening recommendations. How often do you do it? The screening intervals are here. And I can see by the clock on the wall, we've had a bunch of questions, so I'm going to keep going if that's okay, but I will. Okay. All right. I'm going to have to skip a couple of things. The screening intervals for programs of testing. Fecal immunochemical testing every year. That's a lot to ask for people. People think I'm going to get fit tested, but then they stop after a year or two. And if you do it, the program's not working. Cologuard is more sensitive. It should be done every 1 to 3 years. CT X-ray, we don't do this much. Flexible SIG, we don't do this in the United States. They do it in Europe. Colonoscopy screening every 10 years. These are the main strategies are colonoscopy screening and FIT, and some people do Cologuard. The programs above are similarly effective if you do them. The program means over time. If you're going to do FIT, you can't just do it once. That's not going to be as good as colonoscopy. Colonoscopy every 10 years, that gives you protection for a long time. FIT just once doesn't give you that much protection. If you do it over time repeatedly, you've got more protection. And because you've got more chances to pick up lesions, and lesions that appear between the 10-year interval, like for colonoscopy, you can find them with FIT. That's why, you know, a test like FIT can be useful. So if several different programs are equal, then how did colonoscopy get so popular in the United States? And this is sort of fun. And the answer is, there's a date. It happened on July 20th, 2000 in the New England Journal, that colonoscopy became accepted enthusiastically for primary screening. 2 articles say screening colonoscopy finds right-sided advanced lesions. That's further than the SIG. It's on the right side of the colon that aren't found by sigmoidoscopy. And this study use of colonoscopy to screen. David Lieberman, this is the VA study. Here's another study by Tom Imperiali, a trainee, and me and other people that looked at the Eli Lilly screening colonoscopy program. Same question. And we found that looking on the right side, that if you do colonoscopy, here's what you find, and lesions that are on the right will get missed by sigmoidoscopy. How can you find it by sigmoidoscopy? It's because if you see a sentinel lesion, a polyp, then you do a colonoscopy, and that's how it could find things on the right. The thing is, the findings in these studies weren't new news. Everybody expected this. Nobody expected sigmoidoscopy could find things on the right. It finds a few things, but not much. And the authors were doing what Alvin Feinstein, my mentor, said, documenting the obvious. Why are they in the New England Journal? Documenting the obvious. If it hasn't been done, do it. The yield of colonoscopy was what we reported, a 1% chance of colon cancer in screening age people. 5 to 10% were advanced adenomas, large when sigmoidoscopy misses most proximal lesions. And this is what made this accepted. This is new news. Pardon me, this is old news. A guy named Dan Podolsky, who's head of GI at Mass General, and is a basic science guy, not a clinical epidemiologist. You get it?
he wrote an editorial called, and the New England Journal of Medicine is based in Boston. And he wrote an editorial called Going the Distance, the Case for True Colorectal Cancer Screening. And what Podolsky said was, if you do just sigmoidoscopy, it's like doing mammography of one breast. And between that title and that sort of analogy, this made the newspapers. And the editorial put the story on page 1 of the New York Times, where it never belonged. There was no news here to anybody who was in the field, but Podolsky wasn't in the field. He was head of GI, but he's a basic guy. And I'm not going to tell you, we could go over what the New York Times said and so on and so forth. And the point is, nobody ever thought that sigmoidoscopy detects right-sided cancers. And here's the reason why. Previous modeling and recommendations had not relied on the sentinel polyp to detect right-sided lesions, nor did modeling recommendations after 2000. And the test most commonly, recommended should be replaced by a more extensive procedure. The answer is no. This wasn't how people thought, but the editorial, the news report, and subsequent reports had enormous influence. And why isn't colonoscopy the best test? And this is really important. And this is an article written by Mike Pignoni, who trained here in the Clinical Scholars Program. And he's a generalist. He was head of medicine in Austin. At any one exam, colonoscopy is best. But in a program of screening where you do multiple tests over time, long screening intervals like colonoscopy has every 10 years, it admits colon cancers with fast growth rates. Not many grow fast, but we don't really know how many. So a less sensitive test like FIT applied more frequently might, in a program of repeated testing, be more effective than colonoscopy. That's what Michael showed here. And this, he collaborated with people at the NCI. I mean, this was, neat modeling study, but the point is, this is not crazy logical thinking. So gastroenterologists may overstate efficacy. I'm a gastroenterologist. This is a typical 90% statement. 90% of cancers could have been saved if they'd gone for screening colonoscopy. All those figures are available. But the claim of 90% is wrong. It's wishful thinking. It causes unfair expectations for patients and doctors, and it's not necessary. 60%, a more realistic number, is great. That number is based on 5 clinical trials of sigmoidoscopy. It's only half the colon. We can generalize a little bit to the right. There's only 1 reported clinical trial of colonoscopy, and I think I'm about to stop with one neat story here, and I'm going to skip another neat story. Because you guys have seen this. Here's 1 clinical trial. There's only 1 clinical trial of colonoscopy that's been reported. It's so hard to study. And it was reported in the New England Journal in November, and it made the news. You might have seen it. It achieves only 20% cancer reduction and no colon cancer mortality reduction. This goes against the sigmoidoscopy trials. It goes against the fecal immunochemical testing trials. This is a clinical trial of colonoscopy that looked at people in Norway and Poland and invited some to get colonoscopy, compared them to usual care who didn't get colonoscopy. Terrible result. What happened? Here's the news coverage. It says landmark study, benefits overestimated. This is CNN. This is something else. Really interestingly, this was not carried as breaking news in the Times or the Washington Post. And I give credit. I looked, every hour after this thing came out to find out what was breaking news and stuff like that. Because this was a study that reached a dramatic conclusion, but has an explanation for why it didn't, and it never should have been in the New England Journal. This is the comment. This was a weak study, and it shouldn't have been in the New England Journal. First, because it reported results early at 10 years instead of 15 years. And here's the mortality curve. Years, mortality, the 2 groups, very close, no difference at 10 years. It takes a long time in a clinical trial for the benefit of mortality to be seen. If you go out to 15 years, which is their original plan, there might be a difference. But here's the bigger problem. What they did was they invited people to get screened, and only 42% of people accepted the invitation. And the point is, if you don't get it, it can't work. And there's a reason they did it. I think the study was designed in a not good way. Everybody thinks the study was not designed in a good way. And when they saw compliance was so bad, they should have done stuff to increase compliance, because the first thing you want to do in studying something is figure out if it's done like you're supposed to, does it work? And then you can look in the real world and see about who's invited and doesn't come. It never should have been in the New England Journal. And it's a lost opportunity. We might have been able to learn about this colonoscopy, prevent cancer on the right side of the colon. There's only 7,000 people here, so it might not have turned out that way. But this all reflects the difficulty in doing clinical trials. There's a DNA test that I was involved in, the Cologuard test, that I'm going to zip through, and we'll get through and have a few minutes for discussion. The DNA test was developed on the basis of understanding the gene mutations. You remember we've seen this before. Gene mutations here, APC, KRAS, P53, as you go from normal epithelium to adenoma to cancer and so forth. And when I saw this article showing that you can find in the stool mutations, identification of mutations in the stool with curable colon cancers in 1992 in Science, I remember I was standing in the living room up on Burns Place in Chapel Hill. This was 30 years ago. And I remember I was standing in front of the fireplace. Science was a paper magazine that you got in the mail. And I opened it up and I looked here, and, you know, Bernard Levin was one of my teachers. We all knew this was going to come out, but we didn't know what exactly the results were. So I looked at this and I turned to Pamela, who was standing in the kitchen, and I said, Pamela, my career is over. They're not going to need gastroenterologists to do procedures. There's going to be no more research. This is going to solve everything. This is 30 years ago. 6 years later, the guy who founded Exact and invented Cologuard asked me to chair their scientific advisory board for reasons I don't have time to go into, but I sure didn't know at this time I was going to be doing that. Here's their first generation based on the Vogelgram that you just saw. You go from normal tissue to cancer. Here's the gene mutations and stuff. Fecal DNA versus fecal occult blood testing, screening. These are the mutations in the New England Journal. Sensitivity was 52% for DNA, 13% for guaiac-based occult blood testing, which was the main test. And this is a gigantic difference. This achieved the goals of the manufacturer. But by 2004, the goalposts had moved. Colonoscopy had become popular on July 20th, 2000, in part because of an article that Tom and I wrote. And fecal immunochemical testing sensitivity and specificity in well-designed studies were now getting competitive with that test. Exact nearly went bankrupt. They got some money for intellectual property that they sold, and they developed the next generation assay that had methylated markers. It's not mutations. They said, we're going to be agnostic. We're going to take any markers that seem to work. NDRG4 stands for nerve something something. BMP is bone mineral something. You guys probably know all this stuff. But I, to me, these things have nothing to do with any pathway that we've ever known about. And, it could be new biology, but we didn't look at it in the Vogelgram, pathway. And what's neat about this is this says so much for biological reasoning. This is just chance stuff that got discovered and it works. All right, what about the future? We could talk about blood tests and multi-cancer early detection tests, but we're not going to. Other topics, surveillance. I did pretty good through getting through most of this. I don't know if I was speeding. You can pour cold water on me, at the end. But this is the last slide. Here's what we covered. And the bottom line in red is what? Thank you. There's a choice of methods like we don't have for mammography. We've got for breast cancer. We've got some. We don't have anything for ovarian cancer, CA125. We've got transvaginal ultrasound and so forth. But, none of these things work. There's a choice of methods, but you have to do it. If and when you get a colonoscopy, do a good prep. The end. Thank you very much.
Thank you very much. That was great. Well, so like for questions formally, even though
In the chat, please ask questions if you've got any before we head out.
Yeah.
So obviously this would qualify, but for somebody that is definitely under 45, what do you recommend are like the most proven in your clinical experience?
Prevention of colon cancer, don't worry about it. It's so uncommon. If you've got a bad family history, that's a problem. And, you know, people, Tom and I wrote a study too that was in the New England Journal about colon cancer under age 50. It does happen. About 7% of colon cancers happen in people under age 50. And it can happen in, you know, 20-year-olds and 25-year-olds. Katie Couric's husband, Jay Monahan, got colon cancer at age 42. It does happen, but it's very, very uncommon. The biggest thing would be, do I have a bad family history? And you know, you should know that. And mainly that's going to be colon cancer in a first-degree relative at a young age. Or do I have a family history of ovarian cancer and uterine cancer that indicates hereditary non-polyposis coli, HNPCC, or Lynch syndrome? That's uncommon too. And actually, even though, like Rebecca said, it's an autosomal dominant, in 25% of people with Lynch syndrome cancers, they don't have any family history. They're the presenting case. If you have bleeding, if you have an anemia and it can't be explained by other reasons, that, you know, talk about it with a doctor. They might want to do some stool tests. They might want to do colonoscopy. But the basic thing is, it's just not that common. I don't know if that's reassuring or not, or if you have some other reason to be worried, and we can talk about it afterwards if you want, but don't, you know, the most important, don't get obese. And that's not just for colon cancer, that's for all the other stuff that we talked about, like diabetes and heart disease and everything else. But don't overly worry about this. It could happen, but, you know, if we start doing colonoscopies on people when they're 40 and 35 as a routine, we're probably going to be causing more harm than good. And that's one of the things that the task force looks at with age. You know, at some point, it's really rare. There are some harms. They're not very common. Those are considerations too. Yes. Pardon? Yeah. Not with regard to colon cancer. And usually if the eating problem or anxiety or whatever, or, you know, or the eating disorder goes away, your digestive system ought to recover pretty well. And people who are very, very thin from an eating disorder, it can take a while to recover, but the digestive system is really very, very resilient. And none of those things will relate to colon cancer. Does that answer the question?
Yes.
Okay. Other questions?
There's one question from Rebecca on the slide you showed previously on all these mutations without or markers without a biological basis.
Yeah.
If there's no biological basis, why do they come up in these assays? Or is that there's no known biological basis for that pathway-based thing?
This is science. We stumble on things where maybe there is a biological thing that we just don't understand. And, you know, this is what we do in epidemiology. We find associations, and if they're real associations, that says now you guys, the PhDs, got to do the deep dives and figure it out. We can't start by saying something doesn't work because we don't understand the biology, because if we did that, we'd never learn anything. So, you know, this is, and I don't really know that there's been any basic research about those methylated markers, but boy, it sure is interesting, and it gives you humility, because, you know, when Exact got founded by Stan Lapidus, who's a genius engineer, and he developed a pap smear reading thing called ThinPrep and founded Cytek. It's a $6 billion company. His machines are in the Smithsonian. Stan said, this is just an engineering problem. We find the Vogelgram DNA mutations in stool. We got to find human DNA in this bacterial mess, which has its own DNA. This is going to take, you know, $8 or $10 million and be 5 or 10 years. And it turned out to be 25 years and $800 million to do. And we knew the biology to begin with. This means be humble. Be curious and be humble And don't fall in love too much with your ideas. Guys, you listening to me? Rebecca, are you listening to me? You have to be open-minded and just tough as nails with the science and be open to crazy stuff falling out of the sky. But then you got to figure out, does this mean anything, or is it just, is it just garbage? But that's part of the fun of science. Other questions, comments?
I'm not seeing any questions in the chat.
I think we're good to wrap up.
Okay. Hop on with you. Okay, so we would just like to say thank you to everybody for coming. Thank you to Dr. Ransohoff and Rebecca for speaking for us today. And we hope to see you all next week on our final session of Mini Med School about human development. Thank you all for coming.
Thanks everyone for being here tonight. We appreciate your time, and Rebecca and Dr. Ransohoff.